This is part of the Testicular Puzzle story, and it's how does sperm find the egg. So we've taken the sperm into the female tract, but they've got the final stages to do. How do they actually find the egg? So this is the real puzzle. Given the female reproductive tract is so large and the gametes are so small, how does the fertilizing sperm actually find the egg? Where we left things in the last session, what sperm had entered the female tract, they'd got up to the fallopian tubes where they were binding to the luminal epithelium of the isthmic region, and then they become hyperactivated, they change their motility, and that allows them eventually to penetrate the egg itself. But how are they going to find that egg?
This is a very simplified picture. Here we have the fallopian tube, the oviduct, with the ostium up here where the egg is captured from the ovary when it's ovulated, and the egg is transported this way. But coming up from the other way, the uterus over here, here's a uterotubule junction. Sperm come up into the oviduct, the fallopian tube, and many bind to the surfaces of the isthmic region of the oviduct. But we see that sperm are swimming this way, according to this diagram, and the egg is coming this way. How does that work? In reality, the whole structure is very much more complicated. It is not just a simple pipework tube. What we have is here is a cross-section of the oviduct near the uterotubule junction, and we can see that the lumen is quite folded. As you go up into the isthmus, it becomes even more folded, and as one goes to the ampulla, then we see this very highly convoluted internal structure, lots of folds, lots of crypts for things to hide in, for sperm to get trapped in. And here is the egg, the oocyte, the ovulated oocyte, sitting there ready to be fertilized. And here's a histological section. We have the epithelium, which is heavily ciliated. There are a lot of secretory cells, and there's a lot of smooth muscle. So a very highly convoluted thing in which sperm can get lost and in which the egg has to be transported.
Well, in this slide, we're going to look at the movements of fluid within the fallopian tube of a mouse. The fallopian tube has been removed, and we're looking through the wall into essentially the lumen of the fallopian tube. On this side, we're going to see bulk flow of fluid, really caused by peristaltic movements. On this side, we'll be able to see very clearly ciliary movements. Okay, let's look at the bulk movements first, and you can sort of see peristaltic movements happening. Here we go. There we are. Okay, this is the ovarian end, this is the uterine end. There it goes. Now let's look at this side. Lots of ciliary movement. Lots of ciliary movement, moving fluid along the tube. We're now going to look at an very old video looking at the oocyte and its surrounded cumulus mass going down the fallopian tube. It's a very old video, but it's very good. It shows the oocyte with its cumulus mass stained in blue going down the fallopian tube.
An oviduct has been cut open longitudinally with a fine pair of iridectomy scissors.
Let's go back to our simple picture. Here we have the egg coming down the ampulla, coming this way, and the sperm are coming up the other way. So we've got a problem here. We've got transport in two directions. Fertilization takes place up in the ampulla, and eggs don't normally pass down through the ampullary isthmic junction until after they've been fertilized. How do sperm come up here? Is it just by random movements, random swimming? Are they directed up here towards the egg? Many people have talked about chemotaxis. The eggs are releasing something which attracts the sperm to swim up. There is good evidence for chemotaxis in some external fertilizing species, things like sea urchins, where the gametes are all released into the external environment.
A lot of our understanding of sperm and fertilization have come from basic studies on sea urchins. The advantage of sea urchins is they fertilize outside the body, they're external fertilizers, so the gametes are released into the water, they're very easy to look at, very easy to handle, experimentally very manipulable. And it's clear in sea urchins that the unfertilized egg in sea urchins, the egg investments, release a protein called resect, and that acts as a chemoattractant which attracts the sperm towards the eggs.
This experiment is going to show chemotaxis in action. The solution here contains lots of sperm, sea urchin sperm. These are the little red, or the white dots. These are sperm which are in focus. The bigger globules are sperm which are out of focus. So we have a solution of sperm here, or a suspension of sperm. And we'll see them swimming in a minute. In the suspension as well is a chemical called resect. It's the chemical which the sea urchin egg releases to attract the sperm normally. Currently, that chemical resect is hidden in this solution behind a chemical cage. Each molecule is hidden within its own little cage, and it can only be released from that cage by a flash of ultraviolet light. So what we're going to do is to flash ultraviolet light, a little spot of ultraviolet light in this area here. That's going to release the resect so the sperm can see it, and we're going to see what happens to the sperm. And now we're going to see the whole thing in action. Here are the sperm swimming, fairly evenly distributed throughout the field of view. And then in a minute we're going to flash the ultraviolet light in the middle. That releases the resect in the middle, and the sperm begin concentrating where the resect is, where the chemoattractant chemical is. They get more and more concentrated, and if we start looking around the slide, we'll see that they are not accumulating anywhere else. They're only accumulating where the ultraviolet light was flashed, where the caged resect, the chemoattractant that comes from sea urchin eggs, was released.
How does the resect work? So resect coming from the egg, in solution around the egg, binds to ligands on the sperm cell surface, releases inside cyclic GMP, and that triggers a whole range of events, but ultimately it's to do with calcium influx changing the motility of the flagellum.
We're going to look at the motility of the sperm using a sperm which has been loaded with a calcium indicator dye. And when it's exposed to fluorescent light, if there's calcium present, the dye will show the sperm or any calcium as white. We're going to stimulate motility in the sperm by exposing it to cyclic GMP, which will activate the cat-spur channels, and see what happens. So we're going to look at the sperm swimming first, flash of light to release the cyclic GMP. The cyclic GMP then activates the sperm. Let's see what it all does. There's the sperm swimming around in a circle, flash of light, and look, you can see the sperm tail just showing up momentarily. Let's look at it again in just a minute. Let's look at it again. Here's the head of the sperm. We'll kick the whole thing off. There's the head of the sperm going round and round. Flash of light, cyclic GMP. You can see the flagellum. You can see the flagellum. So the spikes of calcium in the flagellum associated with that change in motility.
So the idea is like this. Here's the egg releasing around it a chemical gradient essentially of resect. Sperm swim into this chemical gradient and respond to it by the calcium influx. That changes their swimming behavior, and it keeps changing, and eventually they end up nearer and nearer the egg.
Cat-spur channels seem pretty universal. Human sperm have cat-spur channels, and those cat-spur channels in the membrane of the flagellum let in calcium, and that calcium stimulates the motility of sperm, changes in motility. The ligand for the cat-spur channels, well, there may be many of them. Progesterone is a strong one in humans, changes in pH, prostaglandins as well. All of these things may result from the release of the fluid with the oocyte that occurs at ovulation. So as a follicle ruptures, follicular fluid comes out with probably a lot of progesterone in, changes in pH, prostaglandins maybe as well. And maybe other ligands within the fallopian tube are also important in modulating calcium in these Catsburg channels and modulating motility. One of the characteristic changes which occurs in sperm, which may be necessary for fertilization, is that they change their motility. They become, it's called hyperactivated, with high amplitude asymmetric beating of the tail. Here is a sperm swimming normally, and you can see that these are a series of images of the same sperm superimposed on each other, and you can see that the flagellum is hanging around at the back and beating. But here is a hyperactivated sperm, and we see the very frenzied, higher amplitude motions of the tail here.
So is there chemotaxis in mammals? How does sperm find the egg in mammals? Chemotaxis in mammalian sperm, and it's got 1, 2, 3, 4 question marks. It's arguable. People are not sure. Yes, the hyperactive motility is stimulated by things like progesterone, changes in pH, prostaglandins, etc. But it's not clear that that actually results in a chemotactic response towards the ovulated oocyte and its associated cumulus cells.
If it's not chemotaxis, what is it? Maybe there's a simple physical explanation. Within the fallopian tube there tends to be a flow of fluid from the ovarian end down towards the uterine end. And that flow of fluid is really determined by three things. One is the secretion of new fluid within the fallopian tube, and then there's ciliary beat and smooth muscle contractions, both of which are causing the flow to predominantly go in one direction, from the ovarian end up here down towards the uterine end this way.
And now there's very nice evidence that sperm will swim against the direction of flow. So if the flow is going from right to left, the sperm will be going left to right. Let's have a look at some videos of this.
Here we have some mouse sperm swimming in a very narrow tube. The flow of liquid is going from left to right. On the left hand side we have uncapacitated sperm, and on the right hand side we have capacitated sperm. Both are swimming against the direction of flow. And this is a very nice demonstration of human sperm swimming against the direction of flow and changing their swimming direction when the flow changes. Initially the flow is right to left, then it changes and it goes left to right. As the flow changes, so the sperm reverse their swimming direction. Let's see it again. Flow changes, sperm change. And this is just brilliant. A recent paper showed the track of a live sperm within the oviduct of a living mouse. Both of these upper and the lower pictures are the same sperm taken from two different angles. It's recorded over 107 seconds. The sperm starts at time here and comes along. Here it goes all the way up, swims around and ends up attached to or very close to the wall of the oviduct in the ampulla.
I've included this slide for completeness, partly because it's actually a very nice picture, but also because you may read somewhere that thermotaxis may be involved in sperm guidance. This is the concept that maybe sperm can swim along a thermal gradient up towards the ovulated oocyte cumulus complex. This idea came from the observations that it looked as though the fimbrial end of the fallopian tube near the ovary was warmer than down here in the isthmic region, and maybe sperm was swimming up a thermal gradient. Now that required thermal sensors to be invoked somehow, and some people have claimed that sperm can exhibit thermotaxis, and they may have sensors on their sensory molecules in some ways. I find this, to be honest, quite difficult to believe because of the very small size of sperm to be able to sense the difference between tiny differences in temperature between one end of the sperm and the other seems to me very far-fetched. But you'll read it somewhere. But it is a nice diagram, sort of summarizing what's going on. Sperm coming up here, binding to the isthmic epithelium in the fallopian tube, coming off, binding again, coming off, coming in, bind again. Sperm hyperactivation taking place. From changing the sperm swimming from symmetrical to asymmetrical, especially when they get close to the ovulated complex. Progesterone probably acting as certainly a stimulus to the sperm, and maybe also a chemotactic molecule. And then the hyperactivated, capacitated sperm penetrating through the zona pellucida and, well, the cumulus cells first, then the zona pellucida, and then meeting the egg.
Putting it all together in a very simple way, we have the egg, which is released at ovulation, coming into the fallopian tube, ciliary motion and peristaltic motion taking it into the ampulla. Coming from the other direction, we have sperm, which are swimming randomly, partly within the isthmus, but also against the direction of flow. They keep on binding to the epithelium and come off again, bind to the epithelium, come off again, bind to the epithelium and come off again, until eventually some of them end up in the vicinity of the egg, and one or more will start penetrating the egg investments. So here we have a freshly ovulated egg, surrounded by a little zona pellucida, surrounded by the cumulus cells in their extracellular matrix, which is very rich in hyaluronic acid. Sperm have to get through the cumulus to get to the zona pellucida. The zona pellucida has binding sites of special proteins ZP1, ZP2 and ZP3, which are binding sites of the sperm. The sperm then needs to fuse with the inner membrane or the membrane of the egg, and then we have to ask the question, what about other sperm? We only need one sperm fertilizing the egg. How do we block the other sperm?
Let's go through all the components we're going to deal with now. Starting on the inside, here's the oocyte. It's been released from the follicle by the LH surge. The LH surge causes not only release of the egg and its surrounding cumulus cells, but it also induces the egg to undergo and complete the first meiotic division. And here's the extrusion of the first polar body. The oocyte enters then the second meiotic division, that then becomes arrested in the second meiotic division. It doesn't complete it. Outside the egg itself, we have the non-cellular zona pellucida, a glycoprotein coat. And outside that, we have the cumulus cells, which are embedded in a matrix which is very rich in hyaluronic acid. And this matrix becomes much more hydrated after the LH surge and after ovulation, and the whole cells and this matrix area expand. For fertilization, the sperm comes along, and in the vicinity, we think, of the cumulus cells, it undergoes the acrosome reaction. The acrosome reaction releases a lot of enzymes, which allows it to penetrate through the cumulus barrier, gets it to the edge of the zona pellucida. The enzymes go on digesting their way through the zona pellucida so the sperm can get through. The sperm then binds to the edge of the egg membrane and eventually becomes engulfed inside the egg. And here we see a real life egg. This is one which has been retrieved after ovarian stimulation from the follicle. We can see the oocyte sitting in the middle. You can't actually see the zona pellucida very clearly, but surrounding it you can see a whole load of cells as though they're radiating out. That's why it's called a corona radiata. Further out, the cells get more and more dispersed, all sitting in a hyaluronic acid matrix. This is going to expand. It's called cumulus expansion. Here we are. Here's an expanded cumulus mass with the oocyte sitting in the middle. And this is a completely denuded oocyte. And you can see very clearly now the zona pellucida with the first polar body there and a couple of cumulus cells just sticking on the outside. So now we're going to work our way through this seemingly complex diagram. What we have here is the zona pellucida that's inside the light blue areas. These circles represent the cumulus cells. So you've got the cumulus cells on the outside, the zona pellucida. Then on the inside we have the egg membrane. Here's the first polar body. Inside the egg membrane we've got lots of little granules just sitting underneath the egg membrane. These are called the cortical granules. What we're going to watch and follow is the sperm coming in. They bind to the zona pellucida, they penetrate the zona pellucida, they penetrate through the zona pellucida, bind to the egg surface. You have fusion and incorporation of their sperm head into the oocyte. At about that same time, we're going to have exocytosis of these granules, and that's going to be involved with one aspect of the block to polyspermy. So we're going to follow all of these steps through one after another. Let's start with the zona pellucida first. The zona pellucida has a number of characteristic proteins in there. ZP1, ZP2, ZP3, and some species have ZP4 as well. And this provides a glycoprotein coat all the way around the eggs, allows the sperm to bind to it, and provides a barrier as well, which the sperm have got to penetrate. Let's start with the zona pellucida first. The zona pellucida has a number of characteristic proteins in there. ZP1, ZP2, ZP3, and some species have ZP4 as well. And this provides a glycoprotein coat all the way around the eggs, allows the sperm to bind to it, and provides a barrier as well, which the sperm have got to penetrate. The next thing is that sperm, having penetrated through the zona pellucida, come to lie essentially with their heads on the side alongside the egg membrane, and you have sperm adhesion to the egg membrane. There's a very specific reaction which takes place to allow the sperm to interact with the egg, and that's between essentially two proteins, one on the egg, one on the sperm. And the two proteins are called Juno and Izumo. Izumo is on the surface of the sperm, and Juno is on the surface of the egg. So we're going to look at this in a little bit more detail. We're going to blow this little section up. Here it is. Here's the surface of the egg with the green Juno protein there. And here we have Izumo on the surface of the sperm. And Izumo belongs to the immunoglobulin superfamily and recognizes specifically the egg surface protein. After the binding of the sperm to the egg, we'll see in a minute that the sperm activates the egg with a whole series of ion channels being activated, but with calcium influx being a major one. But one of the things associated with all that is that the Juno on the surface of the egg tends to be shed. And this does two things. One is, if the Juno isn't there, other sperm can't bind, and the other thing is that the shed Juno actually neutralizes other sperm coming in. So this Juno comes off, and any other sperm that come in, the Juno can bind to them and can stop them binding to any Juno which happens to be on the egg surface. So it's one of the ways of preventing polyspermy. So what we have now is that the sperm is at the level of the egg membrane. The Izumo on the sperm has bound to the Juno on the outside of the egg. There may be other binding molecules being involved as well, but these are two very well characterized ones. That's going to cause egg activation, which we'll look at in just a minute. But that egg activation also is involved with the first of the blocks to polyspermy, that is the release of the Juno, which is on the outside of the egg. The Juno all the way around the egg then gets released, or then starts coming off. And that essentially saturates the binding sites of incoming sperm. So incoming sperm, their Izumo is bound to the released Juno, so they don't get close to the oocyte membrane and they can't bind to it because the Juno has been released anyway. Let's have a look at the egg activation. What happens, one of the first signs that you see of egg activation is that the sperm infusing with the membrane here causes permeability changes in the egg membrane, and calcium spikes are induced inside the egg, and those calcium spikes are all to do with egg activation. The calcium release at fertilization seems to be a fairly universal phenomenon, and it's been very well studied, especially in species like sea urchins, where the eggs are very amenable. So here's a series of photos in the sea urchin where the calcium wave inside the cell is being shown up by this wave of light area, and here it is in green. So 1 second before fertilization, no obvious increase in calcium. Here's the point of sperm entry, and from that you get the spread of calcium ions going across in a wave across the whole cell surface until it goes right across to the other side. And now we're going to watch a short video of those calcium waves speeded up in sea urchin eggs. So up here we have a whole series. This is just ordinary light microscope, whole series of sea urchin eggs in a line. We're going to add sperm to them, and underneath we're going to see the calcium waves going across each of those eggs. There we are. Yeah, brilliant. Yeah, even that one. And then they go away again. The calcium release is coming from stores like the endoplasmic reticulum within the oocyte itself, not from the outside. Let's just look at it again. There they go. In the sea urchin, they go across in about 20 seconds in all, each calcium wave. But in the sea urchin, it's only one major wave that goes across, and that initiates subsequent development. We're now going to look at a similar thing, but this time in mouse eggs. Here are the unfertilized mouse eggs. We're going to add sperm to them. The sperm have just been added, and we should see them. Here we are. Here are the sperm. And this is the ordinary light microscope, and you can't actually see much happening. We're now going to start this video over here, which shows what's going on inside each of those mouse oocytes. So just choose one yourself, choose one of these to look at, and we're going to follow it just for a minute or so. You can see that they're all showing calcium waves. The calcium is being shown up by a calcium sensitive indicator inside each oocyte. And when it sees calcium, essentially it changes its emissions and picks up as a yellow signal on the screen. So it's a calcium sensitive fluorescent dye. So what you see is that each mouse oocyte, once it's fertilized, undergoes a whole series of calcium waves, one after another. A calcium wave comes along, goes away. Comes along, goes away. And they're getting slower and slower and slower. The recordings we've just been looking at have actually been speeded up. The calcium oscillations actually start a few minutes after fertilization, and the oscillations themselves go on about every 15 to 20, 30 minutes or so. And they persist until the first pronuclei are formed. What's the role of the sperm in stimulating those calcium oscillations? Well, the sperm delivers into the egg various factors, including PLC zeta, phospholipase C zeta, which seems to be a key initiator of the calcium oscillations. The calcium oscillations do a number of things, but essentially they stimulate egg activation. Egg activation involves two major things. One is the resumption of meiosis. The oocyte had been arrested in the second meiotic division. Meiosis is stimulated again, and the second polar body is excluded or extruded. The other thing that happens is you have an activation of egg metabolism. Early development essentially all kicks off with those calcium oscillations. Let's put it back together again in our general diagram. The sperm has come in, it's fused with the surface of the egg by the Juno-Izumo interaction, and then further fusions take place and eventually the head is incorporated within the egg. But that fusion initiates, after a few seconds or a few minutes, calcium spikes which induce egg activation within the egg. Let's go back to this question now. Why only one sperm? That's certainly what you need from a biological point of view. You need the genome within the sperm to fuse with a genome within the oocyte, and then you'll become diploid. If you have too many sperm around, as in this picture, then the danger is that more than one sperm will penetrate the egg, there'll be too much genetic material inside, and polyspermy will occur with genetic consequences, or failure of development. Well, traditional views to how you can get a block to polyspermy, how do you prevent more than one sperm fusing the egg, really came from the studies on sea urchins, where, as we've said before, sea urchins are a very good experimental model because they're external fertilizers. You can get hold of the sperm easily, you can manipulate how many sperm actually in the vicinity of an egg, and you can see what happens. Because the eggs are fairly large, you can not only look at them easily, but you can look at their electrical potential across the cell membrane because they're fairly big. And the traditional observations sort of gave rise to two ideas about blocks to polyspermy. One was a very rapid block, which took place because at the time of fertilization there seemed to be a depolarization of the egg membrane, and that seemed to prevent other eggs fusing with it. And then there was a later block because the cortical granules, which are just underneath the egg membrane surface, are released, and the release of the cortical granules and the material in the cortical granules changes the extracellular matrix, making it much more difficult for sperm to penetrate it anyway. Here we're going to look at the rapid block to polyspermy, where we're looking at the potential difference across the cell membrane of the sea urchin egg. Here it is before the sperm are added, -70, the sperm are added here, and within a few seconds the sperm, or a sperm, enters an egg, it depolarizes. So now the egg membrane is hyperpolarized, and it remains in that position for a long period of time, and during that period of time no more sperm will fuse with the egg. So we've changed the nature of the cell membrane. And here we're going to see the slow block to polyspermy. So these are two sea urchin eggs, and they're going to be fertilized, and after fertilization will be exocytosis of the cortical granules from underneath the egg membrane, and we'll see that the surface of the egg, or the layer around the egg, essentially forms. This is the glycocalyx of the egg fusing with the contents of the cortical granules, and the whole thing expands, providing a sort of physical barrier to other sperm coming in. Here we go. Here we go. Huge expansion. This is the fertilization envelope. Here's the other one. Do the same blocks to polyspermy occur in mammals? Well, similar, but not necessarily identical. We've already seen that Juno, at the time of fertilization, is released from the egg cell surface, and that may saturate the binding sites on incoming sperm. We know there is cortical granule release, and there's been uncertainty for many years exactly what the contents of those cortical granules do. They seem to change probably the nature of the zona pellucida, making it more difficult for sperm penetration. But one of the things that the cortical granules certainly do contain is an enzyme called overstacin. And what that does is to cleave ZP2. And ZP2 is responsible for binding the sperm. So what you have is the cortical granules release an enzyme which cleaves the binding site, making it much more difficult for incoming sperm to bind to the zona pellucida anyway. And one other factor which may be involved in the block to polyspermy in mammals, the release of inorganic zinc in association with cortical granule exocytosis. That inorganic zinc coming out of the cortical granules may affect the zona pellucida directly to change the ease at which sperm can swim through it. Zinc may also affect the sperm and their ability to penetrate. It's actually called a zinc spark, and it's given the name zinc spark from the very dramatic pictures you can get of the zinc release when fertilization takes place. We'll look in the next slide. We're going to look at the zinc spark in human eggs as they're activated. So here we've got a time course along here in seconds. Up on this axis and with the black line is the calcium. So this is the first calcium oscillation inside the cell. And in green is zinc concentration. And we're going to look down here at what happens. So this is a time course, speeded up. Three different eggs. Look, the zinc spark is going round the eggs. The observant amongst you will have noticed that while this egg and this egg did something, this egg apparently did not. Why not? Because it's an immature oocyte. It hasn't completed the first meiotic division yet, so it's not going to go ahead and be activated by a sperm or anything else. Well, we started off this whole session on this picture, and the question was, given that the female reproductive tract is so large and the gametes are so small, how does the fertilizing sperm actually find the egg? And we've been concentrating on events up in the fallopian tube, both in terms of how the sperm find the egg itself and then what they do when they do get there, and how the first one in actually does prevent other sperm from getting in and providing too much genetic material to the egg and problems of later development. To read more about these subjects, a couple of good reviews here. One is Molecular Mechanisms Mediating Mammalian Fertilization, Development 2019, and this one, Janice Evans, Preventing Polyspermy in Mammalian Eggs, Contributions of the Membrane Block and Other Mechanisms. That was in a special issue on polyspermy in reproduction development. Molecular Reproduction and Development, volume 87, 2020. Well worth looking at both of those to give you a lot more information.